Good evening, everyone. My name's Julian Rayner. My pronouns are he and his, and I have short dark hair, which, to be honest, is more grey than dark these days, with a very short, small beard. I'm delighted to welcome you to tonight's event, and also just to give a quick introduction. I'm a genomics researcher, but I have the honour of being Director of Wellcome Connecting Science, and the incredibly talented people in the public engagement team within Connecting Science played a key role in facilitating tonight's event and also the exhibit that underpins it. It's our goal at Connecting Science to enable everyone to explore genomics and its impact on research, health, and society. And we work with public audiences, researchers, and healthcare professionals to help people from all walks of life to learn, discuss, and engage with genomics. We're based on the Wellcome Genome Campus, which is a beautiful, leafy, green campus just a few miles outside of Cambridge. And it's home to two absolutely world-leading institutes, the Wellcome Sanger Institute, some members of which are here tonight, and the European Bioinformatics Institute. The campus is also sometimes called the home of the human genome, as it's where the largest part of the Human Genome Project, which was the multinational effort to sequence the first complete human genome, was performed. That project, the Human Genome Project, was completed only 20 years ago. It's amazing to think. But genomics, the science, has moved on at an incredible pace since then. And it's very much all around us at the moment. So the news you will have been seeing about COVID variants, all of that information comes from genome sequences, and many of those sequences were generated on the Wellcome Genome Campus. But genomics isn't just about infectious diseases. Genomics can tell us about the causes and consequences of a huge range of diseases. And it can also tell us about how humans, and indeed life on Earth, has evolved over time and how it's adapting to changes such as climate change. One area where it's having a significant impact is in cancer, which is a disease that will directly affect 1 in 2 of us over our lifetimes. And 2021 actually marks 25 years since colleagues at the Wellcome Sanger Institute discovered the BRCA2 gene, which tells us if we have a hereditary risk of breast cancer. In the time since then, in the intervening 25 years, the science of genomics has allowed us to compare the genomes of healthy cells and cancer cells, and that process has really revolutionized cancer research and had a hugely positive impact on survival rates. Some of the people you'll be hearing from tonight come from the Wellcome Sanger Institute's Cancer, Aging and Somatic Mutations program, because as we grow and age, the cells within our bodies multiply, and as they do so, they have the potential to accumulate mutations in their DNA. Many of those mutations are benign, but some aren't, and it's a goal of these researchers and other researchers at Sanger to better understand the role that DNA mutation plays in cell evolution, aging, and development. So I'm delighted to introduce this event and welcome you. It's a particularly auspicious day to hold it, as today is DNA Day in the UK. We're really glad that you're here with us tonight to highlight the importance of this groundbreaking area of scientific endeavor and to celebrate the inspiring work of everyone who's taken part in Flow Cellular and show you what they've managed to achieve together, even during these challenging times. So that's all from me. I'm delighted now to hand over to the amazing Becky Gilmore, part of the Wellcome Connecting Science public engagement team, to introduce the Genome Gallery and the Saturday Museum.
Terrible, muted. My name's Becky. My pronouns are she, her, and I am dark-haired with increasingly red cheeks, it would seem now, even more so after my muting mishap. So it's wonderful to have so many of you here this evening and for you to join us to celebrate the launch of a fantastic exhibition. It's the culmination of a wonderful project that has managed to blossom in, I think we could say, quite challenging times. But before we hear more about it, it's my job to tell you a little bit more about the format for this evening and some of our kind of event housekeeping. So in the next 90 minutes, you'll hear from all of the participants and you will explore their creative outputs. You'll have lots of opportunities to ask questions throughout. We love your contributions, so don't be shy with that. We're using Slido to collect your questions this evening. If you've not used it before, it's very simple. There's a bar at the bottom of the screen which you'll be able to access and input your question. And don't worry if it looks like you're the only one asking a question. You're not. We're gathering them all behind the scenes. My colleague Beth will be working hard to make sure that we cover as many as possible. Just to say as well that tonight, like all of our events, we aim to help everyone explore the science of genomics and what it means for all of our lives. And we respectfully ask that everyone participate in this tonight in a spirit of curiosity and sharing, respecting different views, identities and experiences. This platform is being moderated to ensure that it's a safe and inclusive space for all. So when we first started this project, it was conceived that the resulting exhibition would be staged in our Genome Gallery, a dedicated exhibition space at the Wellcome Genome Campus. And through there, we use a changing programme of displays to explore genomics research and consider and encourage you to think about what the social and personal impact of learning about your genetic code is. Unfortunately, we couldn't be there to have this exhibition, but the online space I think is going to be a fantastic one. But we hope that we can welcome you back to our gallery and our campus experiences before too long. We were really fortunate, I think, for this project to collaborate with the Saturday Museum, who very admirably pivoted to create a virtual environment to bring our project to life when the pandemic meant that we can no longer meet in person. The Saturday Museum is a collaborative project run by artist Lucy Steggles, who's based in London, and George Moustakis, who's based in Athens. It's an itinerant mobile museum exploring alternative models of co-creation and gentle ways to play with existing systems and infrastructures. The Saturday Museum is interested in connecting people locally, nationally, and internationally. It works by starting a flow on a theme. Flows are flexible frames, soft structures, play spaces, indeed our Zoom environment, allowing for something haptic and collaborative to evolve. Flows are triangular. They are a combination of our lived experience of digital dialogues and physical outputs. And really from that, there's no better person to tell you more about this, this whole experience and how this project could be crafted and weaved and brought to life. So it's my great pleasure to flow towards Lucy Steggles, who will lead us through this evening's event. Lucy.
Hi everyone. My name is Lucy. My pronouns are she, her. I've got blonde, well really more grey hair, and I'm wearing gold earrings, and I'm in my kitchen, which is quite apt because that's been the place where we have spent, well, we've spent most of the last year collaborating with researchers and public participants on this project. So I'm going to attempt in 7 minutes to tell you about all the complexity of a project that has happened over the course of a year. As Becky said, I work with George Moustakis, who's currently hiding in the computer, but he will come and join me a little bit later on. So we were invited by the Wellcome Genome Campus to engage in a project where we worked with researchers and public participants to experiment, explore and understand the science around somatic mutation, cancer, aging, and how our DNA changes over time as we age. I went and met Beth and one of the researchers, Alex, once at the Wellcome Genome Campus, which was amazing. It was like a future world. And then unfortunately, COVID happened. And it's a testament to the Wellcome Sanger that they bolted it out, I think, and stuck with it. And we decided that we would try to find a new way of working, a kind of open, flexible and agile way to kind of engage in what was then an unknown. Which is something that in a weird way is quite familiar to George and I. It's quite in the spirit of how the Saturday Museum works. The title of the project is Flow Cellular. Flow, as Becky explained, is something that we put before the words in our project so that we can use hashtags and Instagram to make online archives and galleries. Cellular, we felt kind of connected to the body, living cells in the body, and something that was more akin to sort of digital or telephone networks. As I mentioned before, we decided that sort of in our first and initial conversations with the researchers, baking was a big theme, cooking was a big theme, and we decided that we would take that on board and we would work in the kitchen. So we thought the kitchen was perhaps a shared space between the lab and the studio, and that it was a place where we could use the metaphor of food to explore some of these quite kind of complex ideas around the science. We felt that it was a creative, tactile, messy, sort of playful space. At one point, we even made our own aprons. I'm going to show you our aprons here. So so that we had aprons to keep us from getting too messy. We used everything from sort of bread, fruit, spaghetti, turmeric, milk, mushrooms, and Smarties to explore somatic mutation. We were trying together, I think, to find a common language to make human connections and sort of explore the science and acknowledge that it was both a really complex and quite a delicate subject matter, but also within that there was a lot of laughter. We're hoping, I guess, to find ways to think differently about art and science, to acknowledge the complexity of the research and to find different ways to communicate things, to create spaces to ask questions. Also something that we just wanted to let you know that although we were using food in a kind of playful way, we were mindful that it was a pandemic and we didn't want to waste food. So this meant that you're supposed to be able to eat most things, but even if they are a little bit disgusting, including a bit blue at times. So I just want to tell you a little bit about, so we work together, and what we've produced and what we're launching today is a recipe book and an online web archive of the project that we've created. I just want to tell you a little bit about the structure and the process of the project. So we worked together over a year. There are more hours of Zoom recordings than I'd even like to admit. I've watched a lot of them, they're quite amazing. We called ourselves clusters. I don't really know why we use that word, but each cluster contained a researcher and one or more public participants. Those clusters met for an hour at a time and for about 5 to 7 times over the course of the year, where we played and experimented and cut and chopped and broke and things apart and tried to stick them back together and all sorts of things. We also used to meet as a whole group of people about once a month to come together and see where those shared themes and threads were. And then a little bit later in the project, we did a series of sharing events with Addenbrookes Cancer Patient Partnership Group, sorry, it's a long-winded one there, with Circuit, which is a Wysing and Kettle's Yard collaboration, and with the Science Gallery London. So we did we co-hosted, the clusters and I co-hosted some events to share sort of that wider with a wider public. We also did a couple of events as part of the Engage conference. And we also popped things out into social media, gifts and invitations for people to sort of experiment with us. The project wasn't without challenges. There was challenges in terms of technological challenges, trying to figure out how best to use technology, how to kind of, but I think sometimes this also really helped us because of the researchers are busy people, and it enabled us to kind of take up a lot more of their time and engage with that. But most importantly, now I've got 2 minutes left, I just want to acknowledge that the most important part of this project is the participants. So, and I really want to sort of, and most of this evening is about passing over to the participants to tell you about their experience of the project in their own words. I wouldn't want to put words into their mouth. But there's a couple of people who aren't with us, who joined us at the beginning of the project. So Gabby and Izzy, who were part of Janet and the Wysing and Kettle's Yard group, weren't able to be with us throughout the whole project, but we just wanted to make it clear that they were part of it. And yeah, I just wanted to say that these participants have been amazing. They're unique, brilliant, creative, wonderful, frank, sensitive conversations have been absolutely extraordinary. And for me, it's been a real pleasure to work on this project. I've also been mind-boggled by the personal projects that people have taken on, from 3D modeling to making jams, poetry to photography and all that stuff. So what we're going to do now is we're going to show you some little short films. They're very much fragments. They're little pieces of film and little pieces of cut up audio to try and give you something of the essence of the project. There's no way we can get get the whole thing across, but that we hope that you'll click onto the website or have a look at the PDF of the book or kind of click into the Instagram feed so that we can give you a bit more information. So I've got 30 seconds, but what I want to do is, in the spirit of all of our engagement and all of our projects, there's always been an element of play. So Sarah, one of our researchers, and Lynn have kindly suggested that if you could, throughout the course of this event, think about if you were an ingredient in a hot pot, what you might be, and pop it into the chat. When I get to the end and we do a little summary, I'm going to read off some of those things so we can see what kind of collective hot pot we've made. Okay, so now I'm going to leave you, and we're going to pass over to the first cluster, and I'll see you in a minute.
Okay, so what I've got here is my lovely sandwich biscuits representing your DNA bases. So we've got a sequence like this. Yeah. And obviously the sequence of the bases is sort of important for that's sort of the information for how your body knows how to make the sort of proteins that that do everything in your cells. And you have two strands of DNA, which is kind of the two parts of the biscuit, and these are all sort of paired up the right way in that you've got the right shapes together. But what can happen with chemotherapy drugs or sort of stuff from the environment is you can get stuff stuck, and that means that you can get all this sort of stuff on the side of them, and that makes them the wrong shape, basically. So now when one of your sort of proteins comes along to try and like replicate your DNA, what it does is it's going to be aiming to pull these apart and then replicate it into two different strands. But it comes along and this one is obviously, it's not the right shape, and the enzymes will recognize the DNA sort of by shape and how they fit together. Yeah. This is the wrong shape. So what it's going to do is going to pull that apart, and it's going to get rid of that because that's obviously wrong. And sometimes that works just fine, and what it will do is it will find another part and it will stick the right one back on. So that's fine because that's sort of back how it's supposed to be. But sometimes what will happen instead is it will take this one off that's wrong, and it will put the wrong one back on. So that's obviously the wrong shape. Those don't go together.
Hi everyone, I hope you enjoyed that. So I'm just going to quickly introduce you to Ellie, Ali, Anna and Charlie. I'm just going to read a quick bio for them and give you a brief overview of what we're playing at, and then I'm going to pass over to Ellie to tell you a little bit about her experience of the project. So Ellie is interested in investigating the patterns of somatic mutation caused by chemotherapy drugs, cooking and dancing. Ali has an interest in medicine, Twitch broadcasting and science communication. Anna is into communication in arts and science, dancing, taking film, photographs, and experimenting with 3D. And Charlie is interested in chemistry, yoga, and science communication. Collectively, if you look at our recipe book, the recipe that these guys made was around the ice biscuits, which you saw in the video, looking at different types of mutation that can occur in our genes and really Ellie doing something extraordinary with an icing gun. We've also looked at sticky toffee pudding. Charlie's joined us for a workshop from her rooftop, the aesthetics of chopping board, how far you can push the metaphor of turmeric and milk, 3D modelling, and used a lot of Smarties. So I'm going to pass over to Ellie now.
Hi everyone, I'm Ellie. My pronouns are she, her, and I have a sort of brown and blonde hair, which is tied up at the moment, and I'm wearing a pink t-shirt and gold hoop earrings, and I'm currently in my office. And I'm a third year PhD student at the Sanger Institute, and as Julian mentioned, I work with a group of researchers, lots of whom are here tonight, that are interested in how mutations happen in the normal cells of people's bodies over time, and how sometimes those mutations can contribute to the development of cancer. And as Lucy mentioned, I specifically do research on the sort of different patterns of mutations associated with different things from the environments. That can be things like UV light or cigarette smoke, but I particularly look at the sort of fingerprint of mutations that chemotherapy drugs can leave on the genome of cancer patients' normal cells. And yeah, I've really, really loved taking part in this project and getting the chance to sort of explore some of the concepts from my research with other people. I think my favorite part's been doing some of the sort of practical aspects and thinking creatively about how I can use things like food to demonstrate some more sort of complicated concepts. And it's been really good fun to work out different ways of representing the sort of different kinds of mutations that we study in a physical way, which I think might be helpful for sort of people visualizing them more easily and stuff. And it's been really interesting to hear the sort of questions that the others have had about my work and get some feedback on what parts of my research people have found interesting to hear about and what they think about the things that we're studying, because obviously everyone has sort of personal experience or experience with friends and family with sort of aging and cancer. And as researchers, we really need people to have a positive perception of the research we do, because we rely so heavily on sort of patients donating samples and taking part in clinical trials. So I always really appreciate the opportunity to talk to people about my work and sort of hear what they think about it. And it's been really good fun.
Thanks, Ellie.
Hi everyone, my name's Ali. My pronouns are she, they. I have short, dark brown, curly hair, and I wear glasses, and I have white skin, and I am currently coming to you live from my bedroom in South London. I'm one of the participant group. I am a medical student at King's College London, and so came to this project with some knowledge about cancer and about genetics. And I've come away with a whole new way of exploring cancer and talking about science. I was really interested to hear about how we talk about cancer and disease with food and recipes, but this experience has been really fantastic and has gone in strange places, which all three of us, or four of us in fact, will talk about, and the other groups as well. I have a great apron as well, like Lucy. I have a Fasola apron, and just memories of really great conversations where we explored mutation, we explored change, we explored how small changes can make a big difference to cells and how that can lead to cancer. We explored treatments and how biscuits with icing, as Ellie has said, can be the difference between a healthy cell and an unhealthy cell. For me, it was also a personal journey. I and my family have a long history with cancer. Two of my family members have died from cancer, and I have another who lives with it today. And obviously I knew that going in, but it's been really great to explore that a bit further and to place it into a world which is understandable and accessible. My biggest contribution was the sticky toffee pudding recipe, which you saw me make in that video, which was a recipe my grandma introduced to our family. and a recipe that we sort of carry with us, because we were talking about sort of family recipes being passed down generations, the same as our genes. And Charlie will talk a bit more about that later. It's been a really interesting project, and it's been really exciting to explore this with people who are also really interested from a science perspective and from a creative perspective as well. And I hope you like the recipes that are in our book, because they're really interesting experiments that we got up to. And now I'm going to pass over to Anna to talk about her thoughts.
Well, hi everyone. My name is Anna, and my pronouns are she, her. I've got short brown curly hair. And yeah, it's been great to take part in this project. Before collaborating with Flow Cellular, I personally didn't know anything about aging. And as the project developed, and we had meetings with Lucy and with Ellie, and had the opportunity to do experiments and to do this cool stuff with fruits, I was actually very surprised how such an easy thing as food could be used as a metaphor to understand such a complex thing, and how Ellie, with her great explanations, helped us a lot to understand all of those complex concepts. During the project, I also experimented with photogrammetry, which is a 3D scanning technique where you take like loads of pictures from an object, and then you create these 3D models with the software. So it was also great to experiment with something else and to use an alternative way to visualize, again, that dynamic processes that happen inside our bodies. And now after the project, aging is something that is more present, I think, and has changed the way I think and I perceive my body. And I think, yeah, I will remember how cool it was to create this collaboratively with a scientist and an artist, how much fun it was, and yeah, how it developed without knowing the outcome, that the great outcome we've got now. And yeah, I will pass it now to Charlie.
Thanks, Anna. Charlie, you up?
Hi everyone, I'm Charlie. My pronouns are she and her, and I have long blonde hair and glasses on at the moment. So yeah, I'm a biochemistry student and was also part of this Science Gallery London group cluster. And I particularly remember about using certain foods for metaphors. Ellie was teaching us about how mutations are occurring all the time in our body. And we were asking, okay, then why are we not always getting cancers and stuff? And she was explaining that some of them spread, some mutations would spread and cause lots of effects, and some just would happen and not do much. And I remember Lucy asking, what's something that food-wise, it just spreads when you do something with it? And I happened to have just tried to make a turmeric latte because we were in lockdown and I couldn't go get my usual one. And I remember thinking it spread really, really well and looked quite cool. So yeah, I use that with milk to show that it spreads. And when you put turmeric on yogurt, so the same ingredient on something else, it doesn't spread. And it was a good analogy of how the same thing happening in a different environment has a completely different outcome. And yeah, it was really fun. And unexpectedly, I was really into the aesthetics of chopping boards and seeing which one would look best on camera. Turns out green is not the best. And thanks, I'll pass back to Lucy.
That's great. Thank you, Charlie, Anna, Aless, and Ellie. I just also wanted to mention Anna had an assistant, Rafa, who really helped with the 3D modelling, and I just wanted to bring his name into the space. He's credited in the book and on the website. And if you want to see some more of those experiments, especially Anna's and Aless's stop motion animation, Anna's 3D and Charlie's turmeric experiment, they're all there in the book, and they're all there online. I also think just what Aless was saying about the sort of humanness of the project, and there was a lot of looking at the science, but there was a lot of during the project we thought of everyone as experts. So we had the researchers as experts, but also our public participants are experts, and as Aless kindly put, everyone has a lived experience in some way of aging and cancer. So I'm just going to see if there are any questions in the chat for this group. Beth, are you there? Any questions? If not, I think we're just going to have to maybe expand a little bit. I'm not seeing any, so we'll have to have a look. Just give me 1 second. No, not at the moment. So I think, guys, we've got just a couple of minutes left. We've got about 4 minutes. So I just wondered if you wanted to use this opportunity to say anything else, or something that someone said had triggered something for you, or something else you might want to share. I was thinking, go on.
I was going to say, did Charlie, because we talked before this event about the story of the Granny Boyd biscuits. Do you want to tell the story of the Granny Boyd biscuits? Because I think that was a really... Okay, go for it.
So we were discussing family recipes, a bit like how genes are passed on down the generations within your family. And we were thinking of recipes that have been passed on perhaps from grandmas or great-grandparents. And as I was trying to think of something, I thought of, oh, the Granny Boyd biscuits that I always used to make with my mum. And I'd recently been to see my family in New Zealand, and I do have family with the last name Boyd. And so I just put 2 and 2 together and thought, okay, that's our family recipe. Brought it to the group, showed everyone these great biscuits, simple, 4 ingredients, really nice. And then I don't even know how I discovered that actually the recipe is from Nigella Lawson, and it's like her editor's great grandparents is called Boyd, and it's their biscuits. But I guess that's a good symbol that we are all connected in some way with our genes.
That's brilliant. And I wonder, Ellie, if you just wanted to chip in and say anything else about the experience of sort of using food and having to kind of jump over that to kind of explain some of the science behind.
Yeah, I know. I was sort of surprised by how easy it was in some parts to sort of think of sort of metaphors that linked the two. I remember doing that sort of session where we had all the different pieces of fruit and people sort of cutting them up in different ways, and then sort of asking me if this happened to your genome, like this cut up banana that's been stuck together, like what do you think that might represent? And it was surprisingly easy to sort of think of ways to link it. It was actually a really, really good metaphor for it.
That's great. Thank you, Ellie. Someone has asked in the question if the book will be available to see. I have one of the only current copies of the book. This is it. And I will, there's a full PDF version on the website that you can have a look at. So whoever asked that question, that it is there for you to see. And then there are a number of copies. So if you're very interested, do let us know, and we can maybe arrange to get one sent out to you. If you want to. So I think, guys, we just have 1 minute left. So does anyone from this group have any final thing they want to say before we sign out? No? We're good. Okay, so thank you very much. So I think we're now going to pass over to our next group. Thank you very much.
To share with us your gingerbread experiment.
Yeah. So one of them is still in progress and is a fantastic mess right now. But one of the reasons, one of the side effects of being in self-isolation is that I can't go out and get ingredients. So I had to get the Sainsbury's delivery well timed to basically timed it for this session.
Thank you so much. That's really kind of you.
And so I have lots of gingerbread. Wow. Way too many gingerbread men than I can actually ever hope to consume without
being quite unwell.
I think your neighbours are going to love you. I think they're going to come out, open the door, and there'll be a doorbell ring, and then they'll each have a box of cookies outside their door. I think they're going to love you.
Yeah. So I'm not entirely. This would work a lot better in person, but we'll give it a go.
Yeah. Lynn and I are willing, aren't we, Lynn? Are we willing to participate as best we can?
I trust Sarah.
Yeah, so I think the way that I would envision this working with if we were going to get this to be done by somebody else, is that you have your gingerbread man, then you also need 4 icing tubes, like these are writing icing tubes. And I like the idea of gingerbread men because it kind of like, again, like, coming back to what you were saying earlier, like it's the idea that behind each of the tumor genomes there is a person, and that all of that person's identity, hopes, dreams, not just their epidemiology data, like they're actual people.
Hi, welcome back everyone. So that was Sarah and Lynn. I'm just going to give you a brief bio to Sarah and Lynn. So Sarah's research is into identifying novel and preventable causes of cancer, mutational signatures, the fingerprints of exposures such as smoking and UV light on the genome, and her interests are reading and cats. And hopefully Sarah's cat, Cara, will join us at some point. Lynn is, she's been a big feature of the project. Lynn is, her interests are marmalade making, traveling, meeting people, photographing everything, including lichen, painting, reading crime, dogs, and people watching. Lynn's dog is also a big feature of the project. Sarah and Lynn's recipe in the recipe book is gingerbread people, which you saw in the video, looking at sort of what's inherited and what's environmental, and the sort of complexity of classification and grouping in research. Themes that we've looked at over the course of the project have been cats and dogs, books, spaghetti, dying tangerines, different kinds of mutation, cookie cutters, Sainsbury's deliveries, marmalade, to name just a few of those things. So I'm going to pass you over to Sarah now, who's going to sort of talk a little bit about her experience, and then we're going to go over to Lynn.
So hi everybody, my name is Sarah. My pronouns are she, her. I have long brown hair, which is in dire need of a haircut. I wear glasses, and I'm in my kitchen currently, where we've done a lot of the experiments, and my cat is actually asleep on the sofa, so unlikely to make an appearance tonight. Although she has, as Lucy said, been a feature of most of our experiments. So I've really enjoyed working on this project. In many ways, we've gone on a scientific journey. So we started out by working with different foods to try and explain some of the basic concepts of mutations. So you may have seen in the video the dyed orange, where I've dyed my fingers for 3 days. And we demonstrated the basic concept of mutations by switching out different segments for ones of a different color. And we made our way from those kind of building blocks on over to like my actual research, and we found a really good analogy for my particular research in gingerbread men. So my research focuses on something similar to what Ellie was talking about earlier, so the patterns of DNA damage. So our project, we collect data from cancer patients from across the world, and we're looking at cancers which are very common in some areas and very rare in others, and looking for the differences in their genomes, and combining that with a lot of information about their lifestyle as to try and figure out why some people might be at higher risk of some cancers. So I have some of these gingerbread men here, and the reason why they made such a good analogy is because they represent not just the mutations, which are these icing dots on the gingerbread men themselves, but also because they represent the person themselves. And getting to know Lynn as part of this project has been one of the highlights of it for me. So yeah, and it's been a massive surprise at how many different foods you can use to explain science. So yeah, so the oranges, gingerbread man. I found myself painting spaghetti late at night. It's been a lot of fun. So I'm going to pass over now to Lynn.
Thanks, Sarah.
Sarah, my name's Lynn. I've got long blonde hair, rather crazy. I'm very pink because I got sunburnt this weekend on a windy camping trip, and I'm white, and I'm wearing jewellery made by my favourite jewellery made by my ex-boss, who, when I fell ill, supported me all through a lot of my treatments. So yeah, this project has been amazing, and it was sort of, in a way, it was interrupted by COVID lockdowns and shielding, but actually it wasn't, because it made the whole process really very intimate. And Sarah and Lucy and Beth and I were often in my kitchen together, it felt like, when I wasn't even leaving the house or leaving the garden, or then eventually leaving the village. And but they were coming into my home, and we were having conversations and picking up conversations time after time. And it was great fun. Despite the subject matter, we actually had a lot of fun. And it's been even more weird, because Sarah, I initially would have thought, oh, Sarah and I have got nothing in common, Dr. Moody, very brainy researcher. I come from an arts and education background, and I'm very into visual things, but I'm, not good really imagining science. And my only real contact with the researchers is, sort of seeing them down a corridor, sort of spooky corridor in Addenbrooke's, when I've kind of got lost occasionally as part of my big treatments. And my contact is, that they've been in the background and helped save my life a number of times, and I'm going to need them again in the future, because I've come in as a public participant with direct experience of very rare cancer mutations. So that's kind of an ongoing interest for me, but this has been a much more fun way of kind of exploring it. So yes, and on top of that, Sarah's a cat person, I'm a dog person, so I mean, what could we possibly have in common? But anyway, then we started talking initially, we sort of found books, and I was reading, science fantasy, science fiction, so that was the first thing. And then through our making together, when Sarah was floating, for me, really quite difficult scientific concepts, we were baking and making, and that again kind of felt very intimate, making and all talking while we were chatting and making, which felt very bonding experience, although it was on Zoom, which is one of the strangest things I think I've ever done, trying to cook jointly on Zoom with people that you've never even met. And sometimes we'd be making and dyeing, and the kitchen would be a mess, and we'd have bits of fruit and dye everywhere. But I would be also making jam in the background or doing something. And so it became tradition that every time I made a batch, that I made some for Sarah, and I've been building up a little store. I've got marmalade, I've got special blueberry and raspberry jam, and whenever I've bartered or exchanged fruit or been given windfalls or whatever, I've been making it, and I've got a pot with Sarah's name on, which we haven't yet seen each other, so we haven't exchanged Sarah. And I was at the beach this weekend, and I was thinking of Sarah, and I was looking at rocks, and I brought back Sarah a very fascinating rock with lots of kind of mutations and things, and it just made me think again about our project and things in everyday life that resonated, including trees and lichen. I think so. I just think it was amazing to find out how much we had in common, and I'm not less in awe of researchers, quite the opposite. I think hearing everybody's experience on the project has been, has really, blown my mind. So that was really no surprise, but I'm surprised how much I've enjoyed it. And I think, like Sarah, we both said, getting to know each other and having Lucy helping us along has just been the best thing and the best experience that I could ever have had during COVID as well. So thank you.
Oh, thank you, Lynn. That's amazing. And the jam is amazing. And I think it's important to reiterate that most of the researchers have obviously met each other, but most of the other people on the screen have never met before. And so, to really be able to build these sorts of genuine human connections as part of the project seemed really valuable. We've got a couple of minutes left, guys, but there's a couple of quite important questions. One for Sarah. How do you paint spaghetti?
With difficulty is the answer. I did try dyeing them first, but when you get spaghetti wet, it goes, obviously it's not solid anymore, so that failed. And so I decided to just paint it with acrylic paints, and that worked. That did the trick.
Okay, so that might be one of our slightly less edible food metaphor
Definitely,
projects.
Yeah, I don't recommend that.
And the other question we have, which is, how did the idea of linking aging mutations and food come up? Maybe we can answer that one together. I think I can start us off by saying that at the beginning, it was very much through my initial conversations with researchers in the context of lockdown, especially Janet, who you'll meet later, who was talking a lot about her lockdown baking, and it seemed that people were doing various different kind of food orientated things. But yeah, I don't know if you guys want to add anything else to that.
Not yet. I mean, I think, you know, when we first heard that we'd be using food, I think that was a little tricky, but you know, once you actually started going, actually there were so many things you could do. And as I said, I found myself doing things that I would never have expected.
Well, I think one of Sarah's other experiments, which is in the recipe book, and Lynn's, involves sort of blueberries, cucumbers, I can't remember now, bananas and something else. So have a look for that one. And Lynn, did you want to add anything to that?
No, it was just, yeah, it was interesting chopping up, and I think we learned I learned a lot about DNA, trying to sort of mismatch pieces of fruit and put things back in different places, in wrong places, and imagine. And I think that really sort of brought home the complexity of the DNA strands and you know, how things could go wrong, but also they could be a bit odd but still be fine. And that was quite a weird thing to think about, because I think I always thought mutation had to be a negative thing in science, and in fact that, you know, that's not the case. It can be neutral, it can be a good thing, it can be a bad thing. And so I was coming at it from, because I've, you know, got two very serious ones, I was coming at it from a negative, but actually, you know, these things are going on all the time. And so that kind of, that's really stayed with me, that's resonated. But I don't chop up vegetables and put olives with peanuts and bananas and oranges, and no, don't do that anymore.
Not anymore. Okay, guys, we're going to have to finish up. Just one last final comment. Someone said, what a fantastic bond Lynn and Sarah have made. And my question is, when do you hope to meet in person? September. That's what we're planning.
Yeah, well, I'm hoping you know, things opening up after May. We'll have both had our jabs, so I'm hoping we can meet in a safe space. I come to Cambridge a lot for treatments, so I'm really hoping that we can meet somewhere and do a jam exchange. I'm not sure about meeting the cat. I'm not sure that's going to be allowed. You know, I don't think my dogs would be happy, but I'm sure we can arrange a socially distant space meeting. Jam exchange.
Right, okay. Lynn, we're going to have to go because otherwise I'm going to get told off for the schedule. But thank you both very much, and we're going to move on to the next group.
I'm still quite sticky.
Maybe drop a little bit of flour in.
Yeah, I think you're right.
Oh my God, now it's sort of, you see my dough? It's kind of sort of less sticky, more coming together.
Okay. Whoa.
How that changes anything.
Is this meant to teach us the mutation processes is meant to be chaotic?
Yeah. I think. And a little. Yeah, go on, Jen.
Sorry, I was just going to say the constant need to like adapt and add more flour or add more whatever, if that makes sense.
Yeah, it does. So like the body kind of compensates and it's trying to kind of make it right if it was. And so if there's not enough flour, it will add more flour, but then it's at the point where something goes wrong. There's too much of something or too little of something.
Exactly. I think if you add some of whatever you're using to colour your dough, however much you want, and that's kind of supposed to kind of sort of symbolise a mutation or a change that's happening, and then sort of fold that into your dough.
Oh my God, I don't know what I've done. It's very green.
Yeah, and it kind of just sort of gets everywhere, right? And this is sort of supposed to symbolise, and don't mix it in too much.
Okay.
The point of this is kind of a little bit like there are still regions when this mutation process occurs that look relatively normal, right?
And
Okay.
so normal dough, and for me it's red here, that's sort of intermixed with this sort of red mutations, if you will. And there's now, it's so ingrained that there's no way of getting this out, right? And the more you do to try and fix it, the more it kind of just becomes a mess.
Hi, welcome back everyone. So who we have here now is Janet, Matty and Ken, who are going to tell you a little bit about what was going on there. Just again, the short bio. So Janet's research is looking at structural variants in genes, the 3D genome, what happens to DNA when it experiences significant trauma, and all of those pictures are of her baking, which is quite extraordinary. I think that's just 1 year. Matty is interested in curating art, science collaborations and creative writing, and she's going to read a little bit of her creative writing for us in a minute. And Ken is interested in science communication, discovering science and arts in daily life as an economic naturalist, photography, curating and cooking, and Ken's going to share some of his experience with us in a minute. So Janet, I'm going to pass over to you.
Hi everyone, thanks for joining us this evening. So I'm Janet. My pronouns are she, her. I have long brown hair and I'm here in my bedroom. And so I'm a researcher at the Sanger Institute, like all the other researchers here today. And I kind of look at these sort of big changes in genes. And these big changes tend to happen in cancers, and you'll take a big part that's supposed to be in one place in your genome and it's put somewhere else. And this happens in cancer, but actually it can teach us quite a lot about what's important and sort of what's important in how the cell looks like, what it does, and how it functions. And sort of if we can get to grips with what's happening in cancer, it will help us understand the cancer, but it will also actually help us understand sort of things about normal cells. And as Lucy has said throughout this call, sort of Flow Cellular is focused on the kitchen and on baking. And I think that's been quite nice because it's kind of relatable to everyone. Everybody does cooking. Everybody has at some point probably tried to make a cake. And so it's kind of brought us all together. And actually, I'm, as Lucy said, really interested in cooking and baking. And I've done a lot of baking even before the lockdown, but we got all locked down and there was literally nothing to do. So I ended up doing more and more baking and kind of tried to incorporate this into our sort of Flow Cellular activities. And it was really interesting because the baking is relatable, but the science concepts that we all think about and study all the time aren't so relatable. And it's quite easy for us as researchers to sort of use all these scientific words and not remember that most people have no idea what we're talking about. And so it's been really fun actually to work with Ken and Matty, but also Gabby and Izzy, who aren't here today. And they've just like thrown themselves in there. So it worked out quite well. And we've done lots of these little experiments. So we've been tearing up bread to try and sort of symbolize the chaotic mess that happens in cancer and trying to put it back together, which is kind of what I do in my PhD. And everybody has realized that actually this is kind of hard to do. We've been putting oil and water together and trying to get the oil out and trying to see how mutations kind of get everywhere. We've been frying up mushrooms as sort of an analogy for damage. But actually, as you saw in the video, one of our biggest experiments has been this flatbread recipe. And so we made this flatbread and thought about how cells are constantly adapting because the flatbread is constantly changing. And the dye that you saw in the video is sort of this metaphor for mutations and trying to get mutations out. And turns out it's quite difficult to get this dye out. But yeah, I think it's been a really interesting experience to try and better my communication skills, but actually it's been super easy because Ken and Matty have just been so involved. And I'm going to hand it over to Matty now so she can tell you what she's thought of the experience.
Hi, I'm Matty. My pronouns are she, her. I'm white, I have red hair, glasses, and I've got a Zoom background of the beach. So I've had loads of fun with this project, making flatbread, but also learning really interesting stuff about the science, which Janet explained very clearly, not coming from a science background. And it's been really interesting learning about the potentials of like art science collaborations and learning, particularly as I'm a curating student at the Royal College of Art at the moment. So I was really inspired and decided to write a little creative writing piece about what food means and this really powerful idea of like sharing together, which we really experienced on the Zoom calls together. So I'll just read it out now. I dreamt last night of a time when we can gather again under smells of freshly baked bread, of rosemary and olives, young and old kneading together, flour like a snow flurry blanketing the wooden surfaces. A grandmother shows her grandchild how to flip and roll, to bring air to the dough, to help it grow and change under the hot oven. Do we change too? The grandchild asks, squishing sprigs of rosemary and olives into the bread. Yes, our cells mutate so often. It's part of growing old, aging, passing on wisdom. The dough enters into the hot oven and we wait until it's crispy and brown. We bring it out to share, a communion in a time when the virus mutating isn't causing harm, and we can nourish ourselves without fear. I'll now pass on to Ken.
Hi everyone, thank you for coming to this event. I'm Ken, my pronouns are he, his. I've got black short hair, I'm Asian. I come from academic backgrounds in social science. I'm a doctorate student. I think this project has really added flavor to my life outside research, and I really appreciate it. And talking and chatting with Matty and Janet. I think this project was not only just fun to me, giving me the rare chance of working my computer and cutting board side by side, but also sort of educational. I've learned a lot about cancer and aging since I left my biology textbooks behind in my high school. I was really impressed by how the fundamental processes of both could correlate. I also learned a lot about alternative and accessible ways to communicate science, making the maximum use of household objects, especially during the lockdown, the strange COVID time. To me, reading about cancer and reading aging in my biology textbooks are really learning scientifically on paper, but playing with sticky doughs, spaghettis, oil, water, vinegar, and giving them scientific metaphors is learning by heart. I mean, similar to many of the previous participants, I think this experience really struck me as how many objects we can possibly use in our daily life to give them scientific meanings for us to learn. And to some extent, and at several time points, I even thought about maybe science is just in the eye of the beholder. I just pass the mic back to Lucy.
I'll take it, Ken. I'll take it. Thank you so much. That was great. And I think just wanted to say that Ken is the tidiest member of the project. He's managed to keep his computer clean in a way that nobody else has. Matthew, thank you for your creative writing. That was really great. And Janet, I just wondered if you wanted to say something quickly about the focaccia, because I thought that was a beautiful metaphor that we had in the project.
Yeah, so one of my favorite recipes is this focaccia, rosemary focaccia that I make. And I often do it whenever we're having a family like dinner or when I get together with my friends. And it sort of brings us all together, because I don't know how your family dinners often go, but we plan to eat at like 2 and it comes to like 4 and everybody's starving and there's no food. And I think that's quite a relatable thing, and it brings people together, much like sort of this Flow Cellular sort of has aimed to do. Yeah, I thought it was really great, just this idea of sort of coming together over food. And I think that was a sort of accidental metaphor for our project, that people would come together, share food, and kind of and be able to have these kind of conversations. And I think actually Charlie, who was in the previous group, said that
you know, it was sometimes one of her reflections in our recipe book, which was that it was not only the sort of conversations around the particular science that we were talking about that was interesting, but just how broad things went out. And we went on to cover lots of things, including like the future of food. And Ken, I remember your comment saying, I don't know how I'm going to explain this project in the future. I wondered if you might be able to give us any idea how you might do it.
Well, I think this project to me is really like you start to put the textbook concepts into real life and sort of try to remember it in a way that's really accessible to me.
Lovely, thank you. And Matty, I just wondered, any last words from you? You've got 1 minute.
I mean, it was just how much fun we had in the project and how messy it got, and almost like the space of often doing an activity together can allow people to ask the sort of questions that they felt nervous about, or for people to bring up experiences, which was really nice because it meant we could get hands-on, but also there was lots of time for further engagement beyond just the making of the food.
Matty, I think that's a really important point, and thank you, because it's something I forgot, that we wanted to make these spaces, and I think that was a lot of feedback, that people could ask those questions that sounded a bit silly, but there was an amazing researcher there available, and Janet was great, and the flatbread was quite something. I've never seen so many people online on Zoom at the same time getting so sticky and making multicolored flatbread. So thank you very much, guys. We're going to go to the next group now.
Family would always make these. And so this is also going with the thing we did last time with sifting.
Yeah.
So this is the base cookie recipe that got sifted. And this is the other version. So this is just the same cookie with pistachios, maraschino cherries, and dark chocolate in it. And
wow.
That,
and I wonder which one you're going to eat first.
Well, there used to be 6 on this thing.
Can you hold them up into the screen for us, actually, just because they're so great? So you've changed the one on what's my left is the base, the proper
sifted one,
sifted ingredients as it should be, and then what you've done is you've sort of mixed and thrown in some extra things and not sifted them.
Yeah, exactly. So essentially, well, I mean, you know, going again with this screen being P53, you know, you sprinkle this stuff on it and it doesn't there, and then it turns into. So in this case, the cancer cookie is far more delicious than the.
That sort of it's sort of doing something that's really positive, but it's obviously not, and it's kind of backfiring and doing something that's really negative. And there's something about that in your cookies. It's like, actually you've done the thing that is making a mutation, which is actually causing something hugely problematic, which is probably because they're so delicious, you're going to eat so many of them that you're going to have
like, if you're allergic to nuts. And then my personal connection to those cookies are the fact that the process for making maraschino cherries used to be made from maraschino liquor. So you would soak cherries in a liquor.
Is that the kind of byproduct then of maraschino liquor?
Yeah. But what happened in the U.S. during Prohibition is you couldn't have the liquor, obviously. So a food scientist at Oregon State University, where I'm from, developed how to do it with brine.
Okay, welcome back. So as you can see, Tim and Mike's key question is a maraschino cherry really a cherry? So I'm just going to do a quick bio. So Mike is interested in cryptic crosswords, languages I've got here, travel, making abstract art, and Google Threads. Tim's research is around breast cancer, genomics, and circulating tumors and DNA, gaming, treasure hunts, and slow cooking. The recipe that is the main recipe for these two in our recipe book is the pistachio cookie recipe that you saw, which was looking at the role of TP53, which Tim will probably explain a bit more for you in a minute. Asking the questions about whether a maraschino cherry is really a cherry. Some of our experiments involved fire, lots of things around language, looking at the language of science, things like telomeres and metastasis. I still don't know if I say that word right. Mike did some animation, some poetry. Tim made pesto, did a lot of things with a pestle and mortar, and we learned from Mike that jam and fried bread is something excellent to eat. So I'm going to pass you over to Tim, who's going to explain a little bit about his experience, and then Mike is going to talk a bit and read us some poetry.
Hello, I'm Tim. My pronouns are he/him. And I'm a scientist at the Sanger Institute, where I, in my normal day job, look at mutations that happen in normal breast tissue to see what we can learn about how normal breasts turn into cancerous breasts. So yeah, one thing I quite enjoyed about this whole process is just, you know, the starting at, you know, a list of ingredients or some vague idea of an analogy or a metaphor that I want to make in the kitchen, and then kind of field testing that with Lucy and with Mike, and then you know, in several cases improving upon the original idea as we were actually going. So like, for instance, with the cookie recipe, one thing that, like, after we got into it, you know, the kind of at the end, you had one boring cookie and one very colorful, delicious cookie. And, you know, being able to turn that kind of a step further to think about how, well, really, you know, the tumor is covered in all these mutations. In this case, they'd be cherries and chocolates and whatever. And there's a whole type of cancer therapy that's called immune therapy. And the whole idea is that because the tumor is covered in all these extra bits, it's so much more colorful for your immune system. So if you can figure out how to turn the immune system back on, so it eats the delicious cherry cookie and ignores the boring cookie without any chocolate or cherries on it, then you're able to improve treatment. And so that's the kind of thing that, you know, I wouldn't have ever put any effort or thought into before this whole exercise. But being able to, you know, kind of talk through things, come back to them maybe a week or two later, and improve upon the general analogies. So, I mean, one time, Mike made a comment about, you know, cancer being like one of those trick birthday candles. So you blow it out and then it catches back up. And then, you know, that got me thinking about, okay, well, we can use kind of fire as a metaphor for cancer and cancer treatment. And, you know, sometimes in order to put a fire out, you have to destroy a house. And so that's why you really need to try and catch that fire early in order to be able to get rid of it without having a whole bunch of extra damage. And then one great thing about this whole experience is that Mike is quite the poet. And so being able to, you know, synthesize my babbling into a nice, you know, short little poem about some point that I was trying to get across, and then, you know, seeing sometimes that the point didn't get across exactly how I wanted to, and then working on that was always very useful. So I'm going to turn it over to Mike, and he can tell you about his experience.
Hi, thanks, Tim. My name is Mike. My pronoun is he, him, and I'm currently wearing glasses and a somewhat worried frown. I must start with the caveat that I'm not a scientist. So I hope you'll forgive me if my understanding of some of these concepts might be a little suspect. Early in the project, I started to experiment with some stop motion animation using Scrabble tiles to play with the word metastasis and to investigate how a patient might first learn about it. And we saw some of that in the introduction. But then as we started to discuss various aspects of Tim's work, certain words sparked off trains of thought, and I began to write some short verses. and I'd like to read two of them. The first is about telomeres. They're the little caps that sit on the end of your DNA sequence, and which gradually get shorter each time a cell replicates. And so they contribute to the aging effect. And I actually wrote two poems in memoriam in the style of E.J. Thrib, the celebrated private eye poet. The first poem was broadly sympathetic to the jobs that telomeres do. But then I thought about them again. And I suppose because of my age, I started to resent their lack of staying power, their lack of true grit. So here is that later poem reflecting on what they do. So, farewell then, telomeres. When I was younger, you kept my chromosomes happy. Now, just as things are getting tougher for me, you're giving up. Beware. Do you think you can jump overboard like rats leaving a sinking ship? Remember this, your fate is intertwined with mine. So without me, you cease to exist. Who'll be laughing then, eh? It's literally the nuclear option, mutually assured destruction. Sounds quite a happy one, doesn't it? And then moving on to my second poem, I believe that many cancer patients feel that it's easier to think of their cancer as something that they can envisage, something that they can personify, and therefore something that they can fight against. So when we discuss with Tim how cancers are switched on and off by certain molecular keys, I thought it might be interesting to address a personal poem to a cancer, a new cancer that's just starting to grow. And this one is called To My Tumour. To succeed, little tumour, you'll need the key to turn off a protein called P53, which looks for damage in cell DNA, and finding a rogue cell will put it away. So I hope you don't find that key anytime soon, or I'll finish up singing a different tune. So that's it from me. It's not exactly Percy Bush Shelley, I know, but it's not as bad as some Vogon poetry. So back to you, Lucy.
Thank you, Mike and Tim. That was really great. And I just wanted to add in, I don't know if I mentioned, but Mike also managed to find a kind of Steve Reich music metaphor for cancer right at the beginning for us, and mutations, which was quite amazing. And Tim is our most dangerous participant, without a shadow of doubt. He definitely has the capacity to make me feel afraid that he's going to burn himself down in his own kitchen. But I think there was a really nice relationship that you guys had with language and how you played with language. And Tim, you were very generous in the way that you shared lots of your science with us. So I really appreciate it. I'm afraid we're out of time for questions, guys. So we're going to have to jump on to the next group. But thank you very much. So yeah, next one.
I thought actually, so that it's quite simple. The key thing is really that we're looking at animals that live different amounts of time, like a mouse that lives for 2 years and an elephant that can live for 80 years. And like, why is it? Why does a mouse only live for 2 years? And how come an elephant can live for 80? And what we're kind of finding by looking at the DNA is that they run it, basically these animals are kind of living at different speeds. So I thought a way I might be able to play around with that is sort of with an hourglass concept. Mm-hmm. This is, if you imagine, this is kind of your cell getting mutations over time. And what we find is that basically every animal, once it's got 3,000 mutations, that's kind of the end of its lifespan. We don't know why it's that number, but it seems that all animals, if you look in their cells, at least we're looking at one tissue at the moment. So we're looking at the colon. And different tissues get mutations at different rates. But if you compare the colon cells from elephants, we have tigers, we look at lions, we look at ring-tailed lemurs, naked mole rats, mice, rats, horses, cows. We look at a lot of different animals, and they all, by the end of their life, get about 3,000 mutations in each cell. but that obviously that speed at which they get mutations must vary because a mouse is only alive for 2 years. So if you kind of think of each of these grains of sand as a mutation happening in the cell, if this is human, it's happening very, very slowly. So it takes 80 years for us to get 3,000 mutations. And then I don't know if this will work, but I thought I'll try making it like a faster hourglass.
It's a bit longer than 80 years. Yeah, Melody's not so keen on that idea. Maybe 100.
Yeah. That's
when you're 82, that's not comforting, Alex.
Yeah, there's a huge amount of variation. It's just an average. And that's elephants. Humans are longer.
That's my ultimate favourite piece of video from the whole project, so I hope you enjoyed it. So this is our last group. We have Alex and Melody, who are going to share with you some of their experiences of the project. Alex's research is around animal genetics, as you probably gathered, laser capture microdissections, somatic evolution, paternal age mutations, art, and he says interests in art and illustration. Melody's interested in abstract photography, the story of radiation, kintsugi, and baking. We worked a lot around Alex's research, and we did their recipe in the book is around creating fruit and sort of vegetable animals. We made these fictional animals, and Alex had to sort of speculate on their lifespans, and we had a conversation around them. We made a charrot and a slug cumber, so you might want to use your imagination, imagine what they might be. Other things we looked at was the kind of metaphor of kintsugi, of break and repair, bubbles as a way of talking about sort of the number of mutations. We sowed bananas. Melody made an amazing potato tortoise. Alex made some wonderful illustrations for us that you can see in the book. Melody has taken over Instagram with her photography. Alex also did a wonderful demonstration with an ice cream mold, trying to do a lab demo for me with an ice cream mold, and did a personal lab tour for the group and for Melody of the lab that we can't access, which is also on the website. So if you want to have a look at that, then please do. So I'm not going to take up any more of their time. I'm going to pass over to Alex to tell you about his experience.
Hello, everyone. I'm Alex. My pronouns are him, they. I have white skin, black beard, and I'm wearing glasses, and I'm currently in my living room. And yeah, so this project's been a really unique and surprising experience for me. So in our department, we study how our bodies are constantly changing and evolving as we age. And as a scientist, I love the research that we do, and I'm always looking for ways to share it with others. And I think the things that we're learning about our bodies can kind of change how we see ourselves. But unfortunately, it can be really difficult to convey this to other people. When we think about aging and mutations, there are often many negative connotations that we have. So I really wanted to find a way to kind of make the invisible worlds that we're discovering visible and create a visual language that conveys some of the excitement and wonder of the things that me and my colleagues are discovering along the way. And previously in the past with public engagement, I've often found that a lot of public engagement felt like we were engaging at the public rather than with the public. And so I think this project's been really unique in that respect, and that right from the beginning, we've been engaging with people and communicating with them rather than at them. And I know it probably sounds obvious, but I've really found that the best way to kind of engage and find informative ways to communicate with people is to involve those people in the process of creation right from the beginning. And with these regular meetings with our kind of participants, that's been happening all along the way. And I found that to be a really amazing way of engaging with people about research. And working with Melody, who you'll hear more from in a minute, has been absolutely fantastic. We've done amazing kitchen experiments. We found new ways of talking about science, thought about new ideas for new research projects. And it's been super rewarding and fun, and especially during the pandemic, finding ways to interact with someone and talk about science has been brilliant. I don't want to take up too much more time. I think my last thought is just that it's been fantastic seeing these other groups also and how everyone's brought their own personalities to the projects, and then hearing from them again and seeing how every unique pairing has brought out different ways of communicating about science. So it's been a really fruitful project. And now I'll hand over to Melody so she can talk a bit more.
Thanks, Alex.
Hello, I'm Melody. My pronouns are she, her. I also identify as a tortoise because I'm so ancient and very slow.
we sort of took, Alex and myself, we really took a sort of more abstract view with connecting the art with the science. I don't know whether you know, but Alex is a brilliant artist in his own right. And so we were slightly off the track of we didn't use recipes, for example, and we didn't really use cooking. I think someone mentioned something about do you look at your life with, is your glass half full or half empty? And I just thought mine's bubbling over. Mine's always full to overflowing. And I don't know who it was who thought of the bubbles. We have used bubbles before with Lucy. I've worked on many projects with her, and we have used bubbles. And that was the most exciting thing for me, was the way that Alex explained the life of different animals. There's mutations, how many mutations that each animal has. And when you reach 3,000, that's it. No, I'm going to be the tortoise, because tortoises live to be 120 or something. So that's what I need. I don't need to be cut off in my prime. So that was brilliant. And he explained everything so well to me. We got on really well together. We made... So we did, because I'm very interested in photography, and I needed to do some really, apt photography for this project. And it was lockdown. I mean, I've been shielding, isolating, there's nothing around. So it forced me to be very creative in what I was doing. And it really did me a favor, actually, because I just went off on a completely other track, with my photography, and I'm still out there doing this stuff, and really enjoying it. I found it a very transformational experience. With photography, it actually changed my life and has changed the direction of my life. So I will always be very grateful for that. Just amazing how you have to use, it forces you to be more creative with everything that you think about in a way. I had a lovely afternoon virtual tour of the Genome Campus site. 132 acres it was just an amazing afternoon. The state-of-the-art facilities with the conference rooms, the labs, the data bank storage areas. I had to actually ask Alex very kindly explain to me, what a petabyte was. I had no idea, let alone exabytes, but what was really extraordinary was he said, do you know that we can store whole libraries of data in synthetic DNA? Whole libraries for in something the size of a test tube. Well, that really blew my mind. This is something out of science fiction, if you like, the future of science. It really opened my eyes to, because I don't come from a science background at all. The closest I come is I have a grandson who's taking a chemistry degree, and that's the nearest I've come to anything scientific. But so that was an amazing. I thought, I would love to actually see, be able to do that site in real life, and perhaps we will get a chance to see it over the Genome Campus, which would be really interesting. The other thing I connected with Alex on, I don't know how it came about. We were talking about radiation, and Chernobyl came up, and. When I found out that Alex had been to Chernobyl, I was so jealous, because it's somewhere I've always wanted to go. And he did a field study there into the wild dogs that are roaming around. I mean, it's just phenomenal how the vegetation and the wildlife has thrived, and in that irradiated area. And I just always wanted to go there and to take photographs. Alex spent a long time explaining a lot of things about Chernobyl. It was a really lovely experience. We've had we found we had so much in common, and it's just been absolutely amazing to do all that.
Melody, you've got 30 seconds, quick.
Quick. I just want to say thank you, and Lucy, for pairing you with Alex. It's been wonderful, and thank everybody on the, and especially Beth, who's done all that boring old, other stuff that I can't think of it. I can't think of what she's done, but it's been amazing. Thank you, everybody, and thank you. It's been the most amazing project.
Melody, thank you. That was a really nice place to end. And I just want to say that you guys did this really extraordinary stuff around it. I think Alex talks about it in the book, about sort of trying to find a different kind of visual language for the sorts of science that we're talking about. And Alex, I just wondered if there's any, I'm sorry, again, we're a bit short of time, but any last things you wanted to say?
I mean, I think Melody summed it up pretty well. No, it's just been a fantastic experience, and I'm sure we'll all stay in touch with the participants we've been paired with, and stay free.
Well, our great ambition is to have lunch all together with all of the participants at the Wellcome Genome Campus in September. So just for the audience to know that we have this one remaining ambition to meet in person. But and I'm going to just sign out now, and then we're just going to come back for a quick summary and a sort of debrief before the end. So see you in a moment. We're just at the end of this launch today, and I just want to say thank you. It's a real shame I can't see any of you, but thank you for coming. There's a couple of things I just need to round up with. One, George is here with me on the screen, I hope. George is the other half of the Saturday Museum, and in a minute he's just going to say a couple of words. I also want to say that we have an absent member who Melody alluded to, which is Beth, who's a digital programs curator at the Wellcome Sanger, and she has been amazing. Without Beth, this thing wouldn't be happening. So I just wanted to bring her into the space with us and extend, especially George and I's major gratitude for Beth for doing, as Melody said, all the nuts and bolts and grit. And so I'm just going to pass you over to George to say a couple of words. Then I'm going to read our collective hot pot and a final quote from the Saturday Museum before I pass over to Becky for the final words. So if you can just give us a couple of minutes, that would be great. George.
Hello, everyone. My name is George Mustakas, and my pronouns are he and his. I've got short hair, olive skin, and a mustache, and I'm in my living room in Athens. It's been absolutely fascinating just finding out all about the research and all the work that the Genome Center does. And it's been the first time for me and Lucy to actually work in two different cities, in London and Athens, and creating a visual identity for something like that, whether it's images or GIFs. So it's just been quite a fascinating process, I think, overall. And again, special thanks to Beth and everyone else that contributed. With the recipes, with all the materials from the kitchen and the instruments. It's been quite a valuable, interesting project.
Thanks, George. Glad you're here. Okay, very quickly. If we were a broth, this collective group of people that I can't see would be cardamom, garlic, lemon, sweet potato, unami or red chili flakes, cinnamon, cumin, a glass of red wine, carrot, and flour. And there's a long explanation, but I'm afraid I'm not going to be able to add that in today. Potato, onion, lemongrass, shallots, Mary Piper spuds, veggie dumplings, peppers, dates, dumplings, and olive oil. So I'm just going to read one very quick quote from the Saturday Museum, and then I'm going to pass you back to Becky. So I just wanted to say thank you on behalf of the Saturday Museum to all the people who've been involved in the project. We've loved working with you. It's been an extraordinary experience for George and I to collaborate from our kitchens in Athens and London with such a wonderful group of people, most of whom have never met each other in person. I've been amazed by everyone's warmth, humanity, commitment, creativity, and especially for their capacity to play and experiment with the metaphor of food. We've had a lot of fun, made a lot of mess, and succeeded in generating more questions than we had when we started. It's been a Zoom adventure. We hope this recipe book will live on to get stained, amended, and inspire many more rich conversations. So thank you. Please check it all out. And I'm going to pass you back now to Becky to say a final few words.
Lovely, thank you, Lucy. I really enjoyed that, and I don't know about you, that stew and the hot pot, I think, although a strange mix of things, somehow kind of comes together and makes something that I probably would try at least give a go. I just want to again reiterate your points and say a huge thank you to everyone who has been here tonight, all of our participants for sharing your thoughts, your creative outputs, to our audience for offering your questions and reflections on things. It I think has been a warm and engaging evening. As is always the case, there were more questions and things that we could get through in our time, but thank you all so much for putting them in, and I hope you feel we've captured the spirit of the things that you were asking. I just want to flag a couple of things before you all leave us for this evening. Starting next month, our Genome Late series returns. These are fantastic events bringing together really interesting and thoughtful people for great conversations. Our first is called Art, Science and Lockdown Baking. It's on the 27th of May, and it's going to be chaired by Russell Tovey and Robert Diamant. They're the hosts of the Hit Talk podcast. I'm sure you're very familiar with Russell Tovey's face, even if you've not heard his reflections on contemporary art before. So do join us for that. I think it's a fantastic talk. There'll be a link in the chat to that and to our events page, so you can sign up. You can also sign up to our events newsletter to keep in touch with all the different things that we've got coming on. There'll be 2 further talks in our Genome Late series, all of them tied into the flow cellular theme. If you want to see this again, if you want to share it with friends, you couldn't make it tonight, it will be appearing on our website in a few days' time. So do check back for that, and you can watch it there. And really, before you go, just to say we will have a very short post-event survey popping up for you to participate in. I know everyone asks for your thoughts on these things and you're probably desperate to run off and have a drink or something to eat, but it would be really great to get your thoughts. They do help us shape our future programming, our events. So if you can give us a few moments of your time before you log off, that would be fantastic. So just to wrap up and say, thank you all so much again for being here this evening. To everyone who's been involved, and go and explore the Flow Cellular website. It's a fantastic project, and I hope you enjoy taking lots more from it. Thank you.